So it is funny to I've been studying aging for a long time, and my perspective on it changes as I age. So for example, I always start with a figure like this one. And how many of you have seen this kind of figure before in some talk on aging? If you go to any talk on aging, something like this comes up. So the population of people over the age of 65 is growing, and it's going to continue to grow. This goes to year 2050. So the population of people over the age of 65, and this is in the US, but this is worldwide also, is going to be about 20 to 25% in 2050. So that is a lot of people that are going to be in what we call, the older aged years. And why this is interesting and a potential concern, one reason, of course, is that age-related diseases like Alzheimer's disease are the biggest predictive factor. The most predictive factor is age, of course. And so the prevalence of Alzheimer's disease in the population is also going to grow, as well as other age-related diseases, and just general cognitive changes are going to be more common. Now, most people will not develop Alzheimer's disease. That's something I always tell people when they come into the lab and they're very concerned about their memory changes. Memory changes are normal, and what I'm going to talk about today are normal memory-related changes. But of course, everyone is concerned about developing dementia, and am I experiencing something that is an early sign of Alzheimer's disease, et cetera. And the truth is that most people won't develop it, but as you live longer, your risk increases. So what does change in normal, healthy aging without any signs of pathology, as far as we can tell, are many aspects of cognition. I know not everyone knows what all of those tests are, but they're just various kinds of neuropsychological tests of different aspects of cognition. So some of them are measures of vocabulary, where you just ask people things like, define the word dogma, define melancholy. And others are tests of long-term memory, short-term memory, different kinds of visual spatial tasks. And what we see is pretty clear, which is there's a steady age-related decline across the lifespan that begins at around in the 20s and then continues till the end of our lives. But some aspects of cognition do not change, and in fact may even be improved as we age, and that's primarily vocabulary. So this is a nice, hopeful sort of thing you can hold on to as you approach middle age, as I am now. But you can remember that your vocabulary will be more or less spared or even increase over time, but other aspects of cognition do decline. So just to show this in a little bit more simple way, cognition changes across lifespan, decreases long-term memory, working memory, also known as short-term memory. These decline, but vocabulary for the most part is unchanged as we age. Okay, so cognition's decreasing. What's going on? We're interested in, people who are cognitive aging researchers like myself are interested in what's underlying these changes. What differentiates these normal age-related changes in the cognition from those associated with early, incipient pathology like what you see in early Alzheimer's disease. And surprisingly, that question is still not answered. So we don't really understand very well what differentiates healthy aging from pathological aging, but we are getting closer and closer to this answer. And I would argue that one of the reasons we are getting closer to that answer is because we're using neuroimaging, neuroscience methods in this work. So there are lots of different kinds of methods in neuroscience. Obviously, you're hearing about many of them in this seminar. But the ones that are represented in this figure are ones that we use in humans. So cognitive neuroscience methods like EEG, MRI, PET. These are the three main ones. So the neuroscience of cognitive aging, what this is showing is that over the past couple of decades, the number of papers that have been published using these different neuroscience methods has really skyrocketed. So this is true for for everything. This isn't just for aging, but the number of papers being published with MRI is really gone up. So this happened earlier too, actually. So let me see if I can go back. I think we're.
Maybe under the settings.
I think I just well, we're good now. So the reason I want to talk about this is because, you know, in cognitive aging, I'm one of the people that uses neuroscience methods, but not everybody does. And these methods are very expensive to use, right? So you guys know that, right? It costs a lot of money to put someone in an MRI machine and scan them for 2 hours. So, and it's also more effort. You know, you have to do a lot of work to get someone to be, to screen through and go into the scanner. And I think the onus is on us cognitive neuroscience researchers to really show why it's important to use these methods, because it's a lot more work and it costs a lot more money than it would be to just have people do some paper and pencil tests. So I'm going to hopefully convince you that it's worthwhile by the end of the talk. But I want to just briefly show you this, which I know a lot of people have seen something like this before. Just illustrates the spatial temporal resolution of these different imaging methods. So we don't do a lot of these in humans. Obviously, it would be great to patch clamp a human, but my IRB just hasn't approved that yet. But if we can't use those methods. But what we do is EEG and MRI for the most part, PET scanning. And you can see that on the x-axis is temporal resolution, on the y-axis is spatial resolution. MRI has great spatial resolution, but poor temporal resolution, while EEG has great temporal, but poor spatial resolution. So for this reason, I use both methods in my work to understand both the spatial and the temporal characteristics of age-related memory changes. With regard to EEG and MRI, both have limitations, okay? Primarily the resolution, but there are others as well. So with EEG, what we're seeing here in this spreading current figure here is that because of this inverse problem, it's difficult to know where EEG signals are coming from. So the spread of the current across the scalp makes it difficult to localize where those signals originate. Now, it has great temporal resolution. We know in real time what neural activity, we understand neural activity in real time, but we don't know where it's coming from for the most part. But I will address that a bit later, how we can better determine where signals are coming from. With regard to fMRI, functional magnetic resonance imaging, when we're looking at functional activity in the brain, although we have the ability to determine where things are coming from, where signals are coming from with pretty great spatial resolution, we can't determine very well when signals occur. So that's a big problem. But another problem that is really an issue for when you're studying populations like older people or patients who have had maybe a stroke or have various kinds of cardiovascular issues, there's this relationship between the blood flow response, or vasculature, and brain activity, neural activity and vasculature. And this is called neurovascular coupling. So what we're measuring with fMRI is called the bold signal, which is an indirect method of neural activity, or indirect measure of neural activity. So this is problematic for people who are interested in aging, because as we age, we see changes in cerebral blood volume, cerebral blood flow, most likely the metabolic rate of oxygen consumption. So any of these changes could affect the bold signal, but that's not what we care about when we're interested in cognition in humans. We're interested in this, the relationship between how a person processes an event, a stimulus, and the neural response. So this is an issue that we always have to think about. But assuming we can deal with this, and we have a lot of ways to deal with it, how can neuroimaging help us understand cognitive aging? So I study memory. That's what I'm going to focus on now. And I'll give you some examples of how I think it can help us. So I'm going to specifically talk about associative memory, which is in the domain of episodic memory. So associative memory can be defined, I'll define it here, as the ability to bind and retrieve associations between items. So this kind of memory I find particularly interesting because it's one of the aspects of memory that declines the most as we age, in normal aging. It's also a hallmark of Alzheimer's disease. So for example, if you lose your car in the parking lot, you can't remember the spot in which you parked it. You go to the spot you think where it's going to be and it's not there. That's an associative memory failure. You've forgotten the link between your car and the location that it was parked in. If you forget someone's name, that's an associative memory failure. Or if you have trouble remembering names, that's associative memory impairment. Now, these are common memory complaints in older people, particularly forgetting names. So I like to assess that in the lab, which I'll talk about. But what we are still trying to understand is what's underlying these very common associative memory complaints in older people. In the lab, we have to design tasks to assess memory. And we can't follow someone around in their daily life and measure brain activity, and that would be actually really great if we could do that, and maybe we'll get there one day. But at this time, we can't follow them around and see how they function in their natural habitats. What we can do is invite people to the lab and have them perform these tasks. Has anybody ever been into my lab or another one and done anything like this? Oh, it's really fun. So if you want to sign up, I'll put a sheet out here. No, they're very interesting tasks, we think. So we have people do things like here on the left. You study these face name pairs, and we ask you to decide whether you think that person looks like a Jane or that person looks like an Amy. And then after you've studied a couple hundred of these pairs, you're asked, usually in the same day, but it can be spaced by a couple of days, you're asked to determine whether this is a matching pair or a rearranged one. So was this Jane? The answer would be no, this is a rearranged pair. Was that John, etc. So another example would be you have to make decisions about whether an object in a face or an object in a scene are likely to be found together. So would you likely find that wagon in that meadow? Maybe. Can you imagine that guy riding the zebra? I think that's a zebra, yeah. I don't know, probably not. These are subjective decisions, but the idea is to get people to bind this information together. Maybe just tell me if there's a problem with these mics. So the pattern that we see in performance in older adults across numerous tasks that have been performed for decades, really, is this, which is that memory for items. So asking someone, have you seen this face? Have you seen this object? Have you seen this scene? People are pretty good at saying whether they've seen it or not. And so young and old perform pretty well, on par with one another. But if you ask people about the association, was this paired with Jane? Was this meadow paired with the wagon? That's where we see age-related decline. So you see a disproportionate impairment for associative relative to item memory performance. And this is different from what we would see in Alzheimer's disease, where with advanced Alzheimer's disease, where they've been diagnosed with moderate to severe, where people would just have memory impairments across the board. So they forget items and the associations. What is the cause of memory decline, associative memory decline? So in behavioral work, people have tried to understand this with a variety of manipulations, but it's very difficult, as you might imagine, to dissociate whether, for example, people have trouble processing something about that particular stimulus. So you're aware that there are different areas of the brain that are sensitive to faces and scenes and objects in the human brain. So those are domain-specific regions that are sensitive to those various materials. And we also have areas of the brain, networks of the brain, that are don't care about the kind of stimulus that's being processed. They care more about whatever whatever is being done with that stimulus, whether, which I'll get into a little bit more later, but these kinds of different processes and different brain regions that support those processes are called domain specific or domain general. So one thing we can use neuroimaging for is to differentiate which of these processes or both are affected by age. One problem that might underlie this memory decline would be difficulties in coding events. So it could be that older adults have impaired associative memory because they don't encode the memory as well. Maybe they encode it in a fuzzy way, so they have kind of a vague memory because encoding is impaired. It could also be, it's not mutually exclusive, that they have trouble retrieving it. So the memory's been encoded just as well as it is by young brains, but they have trouble retrieving that memory. It could also be the case that there are impairments in processes, neurocognitive processes, that we would call core memory functions. These are ones that support memory primarily, versus those that what we say, the way we call them is that they work with memory. So these would be functions that help to organize information so that you can encode it, or help you retrieve information that's not spontaneously recovered. And I'll elaborate on what I mean by those in a moment or in a few slides, but those latter processes that work with memory we generally call executive or cognitive control functions. So I adopt this idea or this philosophy that neuroimaging methods can help us to answer these questions. So I'll go through some examples like this one. This is from a study that we did a while ago where we were interested in this question of domain general versus domain specific kinds of functions, processes, and which were affected by age, contributing to their older adults' associative memory impairments. So during study, we had participants look at a computer screen while we're collecting fMRI data, and they studied words and objects, different kinds of stimuli. And during tests, we had them make recognition judgments. So have you seen this word before? Have you seen this picture before? And we also asked them some specific information about the tasks that they had to perform during study. So this is a kind of association that we're testing their memory for. So what we found was that associative memory was impaired in older adults regardless of the kind of material that they had to encode. It didn't matter whether it was words or objects. We've done this with scenes and faces. It really doesn't seem to matter. It's across all categories of stimuli. And if we look at brain activity, and so what this is this is during encoding. People are studying these pictures in the scanner, and we can look at their brain activity and sort it as a function of what later people respond. So I can look at activity during encoding and separate those events that later people remember or forget, and then compare brain activity for those different kinds of events. So this is called subsequent memory activity. And we find that words and objects have different networks of brain activity that support memory for those different kinds of stimuli. And young and older adults show this kind of domain specificity. So in the left lateral prefrontal cortex, we see sensitivity for words, which makes sense if you know something about language lateralization, and the left prefrontal cortex seems to be very sensitive to verbal processing. When we look at objects, we see activity in different regions in kind of posterior ventral temporal cortex. And this is sensitive to object memory. And young and older adults show similar pattern of activity, no difference between the groups. By contrast, when we looked at regions that didn't care what kind of stimuli people were looking at, all of those regions cared about is whether that was going to be a remembered or forgotten event. We see older adults under-recruit some of these areas, these domain general areas, like in the left dorsolateral prefrontal cortex, amongst other regions. So if we look in this study and others, we've done this many times, in regions that we would call core episodic memory regions. So we call them this because, for a variety of reasons, but one reason is that this network of regions, like the hippocampus, parahippocampal, gyrus, some of posterior parietal areas, seem to be, they're functionally connected. Some of them are anatomically connected as far as we can tell. And they seem to be integral for memory functioning. So patient HM, how many of you have heard of patient HM? Okay, so patient HM had bilateral, well, he had his bilateral hippocampi removed, and he was left with a profound amnesia. So these regions are essential for episodic memory. Now, in these areas, we don't see age-related change in many studies, time and time again. So young and older adults equally recruit these kind of core episodic memory regions. And by contrast, we see lots of changes. Well, we see the majority of age-related changes in the prefrontal cortex, bilaterally, lateral prefrontal cortex. And we've shown this in both when people are studying and later when they're retrieving, trying to retrieve memories. We see under-recruitment by older adults in these prefrontal areas. And if we look at anatomical data or structural change over time, this is from each of these bubbles on each of these graphs is one person's brain volume. And this is not a longitudinal study. This is a cross-sectional study of a large sample of people from ages 20 or so to 80s. And you're looking at the volume in the lateral prefrontal cortex, primary visual cortex, and the hippocampus. And although there's decline, there's atrophy across the lifespan in most of the brain, some areas are spared, like primary regions, visual, auditory, somatosensory. Where you see the most decline is in the lateral prefrontal cortex, and this has been shown many times. So this idea of the prefrontal cortex showing the most atrophy across age or across the lifespan, together with findings like what we've shown in our imaging studies, where we see most of our changes in age-related changes in the prefrontal cortex, together have been put forward in this hypothesis called the prefrontal cortex, or prefrontal hypothesis of aging, which suggests that it's largely changes in the prefrontal cortex that underlie normal age-related cognitive change. When we talk about pathology, like what we see in Alzheimer's disease, the story is quite different. With early Alzheimer's disease, or mild cognitive impairment, which some people believe is really early Alzheimer's disease, you start to see pathology first in the medial temporal lobe. And as the disease progresses, that pathology, amyloid burden, for example, spreads throughout the brain. This is not what you see in normal aging. But what's problematic is that if we waited until someone had severe Alzheimer's disease, obviously there are going to be differences between a healthy person and somebody with Alzheimer's disease at that stage. But it's not great to wait until that happens, because the idea is to try to differentiate healthy aging and pathological aging early enough. And this could be as early as 40s, because these changes this pathology can occur quite early on. Even in your 20s, there's evidence that people can have plaques in their medial temporal lobe. Just to put that out there in case you're in that, I'm no longer in that age range. But the idea is that early on, very early on, we may be able to detect differences between healthy people and people who have incipient pathology. And the cognitive differences between MCI and healthy older adults are very much harder to discriminate than in severe late-stage Alzheimer's. So if we're trying to understand what the cause of age-related memory changes might be using neuroimaging, we can know what the cause is, and there probably is not one cause, but we have a little bit more information. So we seem to support this idea, our data support the idea that it's largely the domain-specific processes. They don't really care about what kind of specific materials are being processed, but more about what the person is trying to do with this information. Are you trying to encode it in in association with other information. I'm not wording this very well. When I get to cognitive control, I'll talk more about that. But it doesn't seem to be at least the domain-specific perceptual processing regions that are affected by age, accounting for memory impairments. We show data that supports the idea that older adults have difficulty encoding distinctive events, but they also have difficulty retrieving events. We know this from our neural data. And we don't, I don't know. Does this happen to anyone else? Probably it's just me. I'm sure. most definitely. And in healthy older adults, we don't see many changes in core episodic network. Most of the changes that we see, and I argue this quite often when I'm talking about my work, is that most of the changes we see are in what we call cognitive control or executive control. It's kind of the same, different term for the same thing. Processes. And these are processes that help us integrate information to facilitate memory encoding or help us retrieve information, particularly when it's not effortlessly recovered. So I'll give you some examples of that in a minute, of those kinds of processes. These are what we think are largely affected by age. The problem is that in trying to understand cognitive control is not one thing. And we don't know which functions are affected by age. So this is really what we're spending a lot of time trying to do in the lab now, is disentangle the different prefrontal contributions to age-related memory impairments. So the prefrontal cortex in humans is large and heterogeneous. And we know this based on work from human patients, work from monkeys. We know this from different kinds of patients who have damage to the prefrontal cortex from stroke or aneurysm. So, and of course, neuroimaging work. In aging work, most researchers have not tried to disentangle these different prefrontal contributions. We've sort of treated the prefrontal cortex as just this box, and it's affected by age, and that's really kind of all we know. But we can try to disentangle the different functions, and if we look at, for example, volume loss across the lifespan, this is a prefrontal volume in different prefrontal ROIs, regions of interest. And you see that if you just look at the slopes, the prefrontal cortex is differentially affected by age. So not all of these regions are exhibiting decline to the same, or atrophy to the same degree, some more than others. This isn't looking at cognition, this is just looking at volume loss. We know from, and this again from monkey work and human patient work and imaging, that the prefrontal cortex is hierarchical, or at least some models suggest that. So what I mean by this is that some regions of the prefrontal cortex, particularly those towards the rostral end, or the anterior end, are thought to be responsible for high order functions, processing information at a more abstract level. Whereas more caudal areas of the prefrontal cortex are more interested in concrete processing or low order functions. So just to give you an example of a task that someone might do and show this kind of differentiation, regions in the caudal PFC might support processing of, when I ask you to determine whether this is a red apple or a green apple, just a very simple kind of color judgment about an object. Regions of the prefrontal cortex are important for that, caudal regions. If I ask you to compare the color between two apples, then that's a higher order process. And then if I ask you to compare the color of 5 different apples or 5 different objects, or memories of apples that you've, there's an apple in one of my slides, I think that's why apples are in my brain, but, or I'm hungry or something. But if I ask you to make these kind of multiple sort of comparisons, that's a higher order process. And we see that different regions along the rostral caudal axis are important for those different kinds of specific functions. So these are different models of the PFC. I'm not going to go into all those details, but I just want to mention this because it's going to come up in some of the I'm going to present. So what we're trying to do in my lab these days is disentangle the cognitive control contributions to associative memory and see which are affected by age or disproportionately affected by age. And the way in which we do this is by manipulating the demands on these different cognitive control functions through our tasks, experimental manipulations. Why we do this in the first place, like you might wonder why anyone would care, is because we feel like that if we can understand better which are affected by age, which specific prefrontal regions, which specific networks and the functions that they support are affected by age, we can develop more sensitive interventions. So one intervention would be just a cognitive intervention where you have people learn how to use a particular strategy better. That's just a simple one that people have done a lot. Alternatively, you could use method like brain stimulation and stimulate particular areas of the prefrontal cortex that you think are underutilized by older adults, and perhaps that could lead to amelioration of cognitive impairment and memory impairment. We're not there yet because we don't understand exactly what regions we should stimulate. So that's part of the reason why we're doing this work. So here's a study, a task that we've used very recently where we're trying to understand these different cognitive control contributions to associative memory. And here's that apple. So you study these colored objects, and you have to determine whether this is a likely color for the object, and the answer should be, who thinks yes? Okay. Hopefully not. It's no. Most people say no. Every once in a while somebody says something like yes, and you just wonder what's going on with them. But the answer is no. And here your attention is directed to the color-object association. In the bottom example, the item-directed task, you have to decide whether that object would fit inside of a shoebox. These are really common sorts, what we call orienting tasks in these kind of memory experiments. And it's only in the association directed where people are attending to object color associations. What we're reasoning here is that in that association directed task, memory for the object color association is dependent on making multiple comparisons between objects and colors that I've generated. So in order to decide whether that blue apple is a likely one, a plausible one, I have to generate from memory multiple exemplars from my own experience. Have I walked through Whole Foods and seen a blue apple? I mean, maybe, because they have all kinds of colored vegetables and fruits there, but probably not. So I have to run through all the examples and compare them with what I'm seeing. So this is tapping into what those higher order functions that are supported by rostral PFC regions. By contrast, at retrieval, so this is memory, this is when we're scanning while people are trying to recover memories. We have this task where people decide whether this is an old or new object and whether the color matches what they'd seen the object in before. The answer here is no, it's different color, but same object. Here, when you have to recover color, the object color association, but your attention wasn't directed to it, it's more difficult, of course. And one of the things that people do is they have to decide whether what they've recovered, the memory that they've brought to mind, like the blue apple, matches what they're seeing on the screen. And that matching between what I've retrieved and this stimulus is a lower order process. So this is happening at different stages, but we're separating some high order from low order functions. We show here in memory performance, so on the y-axis, 1 would be 100%, just to give you an idea, and 0 is chance. So people aren't performing at chance, but older adults are performing a little bit worse than young adults, no surprise there. But they're showing the same effect, which is that when your attention is directed to object color, your memory for object color is better. So that was kind of nice that everyone showed that pattern, but older adults to a lesser extent. But with regard to the imaging, what we find, and this is during encoding, so again, looking at activity in the brain while people study and separating it as a function of what they later remember or forget, you see that regions in the rostral prefrontal cortex support successful encoding of object color associations. And this is for the association directed, is this a likely color condition relative to the item directed condition. And only young adults show this. So this was, to us, pretty interesting. It suggests, at least in this experiment, this one example so far, we have a few others, but that older adults are not recruiting these kind of high order rostral PFC cognitive control functions when they're encoding. So how we would address this with behavioral data alone, I don't know. So this is an example of where we think the neuroimaging is helping us better understand what's going on, causing those memory impairments. During retrieval, when people are asked to make those color discriminations, is this a one you've seen, is this the same color or not, we find that more caudal PFC regions, like this ventrolateral PFC area, is equally recruited by older adults and young adults, rather. So in this direction that we predicted, where you're having trouble making that color discrimination because you didn't bind color to object or apple, for example, very well. And so you have to decide by comparing what you've recovered with the one you're seeing if it matches or if it doesn't match. So this is a lower order kind of cognitive control process. Okay. What we often do in the lab is we will run experiments, fMRI experiments in a sample of people, and then we'll run a parallel EEG experiment because, as I started off saying, we're interested in using both methods to understand age-related memory decline. And one of the things that we can do is comparing across experiments, which is not ideal, and so we're doing something new now. But what we are trying to do is understand if we can see a similar pattern of activity with EEG that does a couple of things for us. One, it gives us some information about temporal the temporal characteristics of this cognitive process. And two, it helps support the idea that the changes we're seeing with imaging, with fMRI, are neural in origin and not perhaps some to us uninteresting cerebrovascular related change, or reflecting that. So this is an example of when we did just that, where we ran a parallel EEG experiment. And this is just showing one electrode. And what is being depicted here is something called an event-related potential, which maybe a lot of people have heard about. But this is an average of EEG activity over many trials, many events. And we're comparing those two kinds of tasks, whether you attended to the color or made the shoebox discrimination. And this is during memory recovery, where we find this kind of very sustained pattern of activity that shows the same direction, greater activity for item-directed than association-directed, over these lateral kind of prefrontal scalp sites. Now, we don't know what region these are coming from because, again, the inverse problem precludes us being able to make those kind of conclusions, but we're seeing the same pattern of activity, and now we know something about the time course. And although it may not seem like it, something that occurs 1,400 milliseconds after I see that object and make my memory decision is quite late in terms of cognitive processing. So this is happening well after that memory has been recovered. Now I'm just in kind of some evaluation stage. So it gives us some nice temporal information that we don't have with fMRI. Just mention this briefly. We do a lot of ERP work in the lab, but we also look at neural oscillations. So we showed the same kind of pattern of activity. This is in a different study in alpha and beta band, in alpha and beta frequencies. And the reason I mention this is because we're starting to do real-time EEG in the lab. I'm going to talk about that at the very end. And although you could potentially use ERPs and kind of diagnose them in real time to determine whether somebody's likely to remember or forget something, neural oscillations are a little bit more robust. at the single trial level. So this was nice for us to be able to see this because we're going to be using neural oscillations and looking at them in real time to predict memory performance in people. Okay, so the data that we've collected so far, we think, suggests that older adults can recruit caudal PFC areas, but not rostral PFC areas, or at least they don't spontaneously do that. But what we don't know still is whether these rostral PFC regions and the operations they support are just unavailable. It could just be that older adults, despite having the same instructions as young adults, are just doing something different in the lab. If you've ever conducted an experiment with humans, or I mean, I don't know, non-human animals, they do different things, I suppose. Some perform well, some of you guys can speak to that, and some don't, and you don't know why, right? Does this happen, or am I? Yeah. Okay, it's even worse with people. So you get one person who comes in and performs the task in the way you think they should be, or at least you think that's what they're doing. And then somebody else does something different, and you think, how the heck did you come up with that? You know, they're just, people are creative. And so you have people that bring these various kinds of ideas to your experiment, and maybe older adults, on average, just aren't recruiting the operations supported by those various prefrontal regions. So the way in which we can better understand whether a region is just under-recruited or unavailable, there's potentially two different conclusions, is by using other methods that assess integrity of regions, like something like perfusion MRI or PET imaging can assess this to a better, so we're using some of these methods as well, but I'm not going to talk about those. The last couple things I want to mention are that what we're doing now is bringing together EEG and fMRI in a way where most people haven't done this before. So typically you have EEG folks and you have fMRI folks, and they don't, I mean, we talk to each other sometimes, but we really don't come together and do the same, collect data, EEG and fMRI data for the same people. But we are starting to do that now. And the reason we're doing that is because we can apply newer methods to better localize EEG signals using a person's own functional MRI activity. So as I mentioned before, if we can do this, we can test temporal predictions about different prefrontal areas. It gives us more information about these different cognitive functions and which are affected by aging. And we can also say more definitively that these fMRI signals are really neural in origin and not, because the EEG doesn't have the same issues, the same limitations with regard to that neurovascular coupling. That's just not an issue for EEG. So there are a few reasons why we're bringing these data together. And the way you can do this is you can use, there are different approaches, but the one we're using is to collect EEG data with a high density electrode array of 128 or 256 electrodes. So you have all those electrodes on your scalp, and you measure EEG activity for an experiment outside of the scanner. We don't do it in the scanner because that's not great. There are lots of problems associated with artifacts that affect the EEG, and actually EEG that affects fMRI data quality, so it's better to do them separately. And if you have EEG data for a person and you have MRI data for a person, you can do a kind of modeling where you use what's called forward modeling, and you can say, based on this person's fMRI activity, the regions where the brain seems to be responsive in a particular way, I'm going to use those to inform my EEG source localization. But it's a very cool kind of newish method that's being used in a few labs out there. So I'm on sabbatical this semester. In a few weeks I leave, and I'm going to be, I'll come back hopefully imbued with knowledge of how to apply this in the lab. But this is a very cool method, because what you can start to do now with much better accuracy than we've been able to do for decades, is you can decompose processing stages of memory for different brain areas. You can track in real time, in EEG time, millisecond resolution, what these different brain areas are doing. So this is from a working memory study where they were looking at perceptual evaluation, maintenance of information, controlled access, retrieving that information, and they could track these different prefrontal areas. So I thought this was very cool, and I'm hopeful that it will work for us as well. So just to wrap up, I think I've convinced myself, at least, that neuroscience can make valuable contributions to our understanding of cognitive aging. I've showed evidence that neuroscience methods can disentangle age-related changes in various stages or domains of processing, encoding versus maintenance versus retrieval. We looked at dissociations between domain-specific and domain-general kinds of processes. We can look at neuroimaging data, and it can help us understand whether the cognitive changes that we see in a person are potentially related to incipient pathology that just hasn't reached like kind of full-blown Alzheimer's stage or development, or just normal age-related changes in cognition. And we can also use neuroimaging to potentially enhance learning in real time. So this is where we're starting. This is my new pet project, where we're trying to use EEG with a brain-computer interface to enhance person's learning as they learn in real time. So this is my picture of this, my figure, where we might have people study those face name pairs, like I showed you before. We're recording EEG from a high-density EEG array. We take those EEG signals and we look at different frequency, different oscillations across the scalp, and we can apply pattern classification analyses, which maybe some people know about, using like machine learning-based methods to determine whether that brain state, is what we call it, is more reflective one that's likely successful for encoding or unsuccessful. And if unsuccessful, then we can use our brain-computer interface approach to have people relearn information that they would otherwise forget. So we're starting to work on this now in the lab. It's one of my favorite things, because it's new. Your favorite thing is always the new thing, and then you realize that it's full of problems too, and then dislike that one. No, but right now I think it's perfect and it's the best thing ever. But no, I'm very excited about this. So we're starting to do this now. And now machine learning is just sort of really exploded to this very exciting level where I think that we can actually do this, whereas 5 years ago I wouldn't have thought we could. But so that's it. And I just want to thank everybody who does all the hard work in my lab, particularly our dog mascot, of course. He's the most important. No, but this is all my, my lab, and I don't think any of them are here, but I didn't tell them I was going to be here. So. All right, thanks so much, guys.
Oh, wait. Annabelle has one over there.
Hey.
You mentioned at the beginning, but I didn't really talk about, is that the cerebral blood flow and cerebral blood volume change in aging. And it looks like a lot of comparisons you're doing within the same subjects, you can control for that. But how do you deal with that?
Yeah, so that's a good, there isn't just one thing that we could do. So, well, one of the things that we can do, which is very new really, is to calculate a person's cerebrovascular reactivity, and you can use that to correct their task-related bold response. You can do that with something like you can induce hypercapnia and a measure of cerebrovascular reactivity that way, but that's not very pleasant for older adults. So one of the newer ways that people are doing this is to use heart rate and resting state bold, and then correcting with this cerebrovascular index your task-related bold. But typically, people have largely ignored this problem, if I'm being completely honest, including myself, and because it's hard. And what we typically have done is only tested the most healthy older people you can imagine. I will probably never be like this myself, but they take no medications. They are very, very high functioning. Many of them are alumni of Georgia Tech, which is not your typical, person over the age of 60. And so they don't take a lot of medications they don't have hypertension and that sort of thing. But yeah, so there are many others, but I'd be happy to talk with you about those later, actually.
So I have a scientific question, but also a sort of sociology question. Maybe I'll ask a sociology
Okay.
question first. You were making the case, which I think probably many people in the audience here would think, well, that's a foregone conclusion, that neuroscience serves should serve a prominent role in studying cognition. So what I can't quite get my head around is like, what else would there be? So that's one.
Right. It's so funny. That's like the opposite of what I usually get at just cognitive conferences. Well, right, so you can be very clever with your experimental design and try to disentangle different cognitive processes with your tasks. And there you're trying to get a sense of where people have trouble and where they don't. This is all just kind of what we call behavioral manipulations. Yeah, but I mean, I think without the behavior, the neuroimaging to me is not interesting. So I'm only interested in when I can bring them together. So we need both, but.
So maybe that leads really nicely into my second question, which is more scientific, which is, you said something really intriguing about that maybe, certainly in solving behavioral tasks, different individuals solve them in very different ways, right? Just watching a kid solve a problem versus different kids or kids versus adults or whatever. But I'm also intrigued at the idea that during aging we may be solving the same problem that we solved 20 years ago in a different way with a different pathway. And so there's sort of a non-uniqueness problem, right? There's different circuits and different brain regions that could get engaged to solve exactly the same problem, in which case that's pretty daunting, right?
Totally, yeah, completely daunting. So what we have to do in our experiments is try to constrain them as much as possible and instruct people as much as we possibly can to do a particular thing. Because otherwise, we know actually, and this is from behavioral data only, we know from decades of data that older adults, if you ask them, remember these words, they will not use the same strategies that young adults do. So what would you guys do if I said, remember this list of words, real words, but
I think of pictures of those things.
Okay, imagery, yep. Sentences maybe involving the words. What you shouldn't do is repeat them. Rote repetition. That's a horrible strategy, but that's what a lot of older adults will do.
So I'm not old, is what you're saying.
No. But it is something that as we get older, we use these strategies that aren't very effective. So if you looked at brain activity, you might say, oh, look how little the older adults are recruiting this network. But actually, you haven't put them on equal footing. Older adults can do the same thing. You just have to ask them, do that. And then, oh, look, the brain is doing the same thing in young and older adults. But still, it's a daunting problem.
Other questions, comments?
So how quickly can you analyze the data online and give them feedback? So you want to do it on a different trial? Would you be able to do it so quickly as to extend the picture presentation in real time until they reach the desired state?
That's a good question. So maybe. I wouldn't do that because people have worked out how repetition learning works, and the best method of doing it is actually with spacing. So if you repeat things, 1, 2, 3, 4, 5 right after another, that's not going to lead to better memory as much as if you space them out. So for our perspective, it's not a huge problem, but with how real time can we get, that's a good question. So I just met with somebody actually last week where they They can do this, they think, in a matter of like tens of milliseconds, using the graphics card. Do you guys know about this? Yeah. So that was news to me, but that's something we're going to try out. But to me, I don't know if I would really be the best user of that method or that technology, but it exists, so it can be done.
Other comments, questions? Okay, well, let's thank Audrey.